Hello, my name is Navina Lilie and I'm a PhD student at Bielefeld University in Germany. Together with my colleagues, I investigate the effects of sperm competition risk on behavior and hormones in male zebra finches. In sexually reproducing species, individuals have to compete for existing mates in order to reproduce and maximize their fitness. Sperm competition selects for precopulatory competitive male traits such as courtship and aggression. Thus, sperm competition risk is a key dimension of the male social niche to which individuals are expected to adaptively adjust. This we define as male social niche conformance. But actually, experimental evidence showing that males plastically adjust their phenotype to sperm competition risk is relatively rare. Hormones play a key role in regulating behavioral variation within and among individuals and are known to be highly sensitive to changes in the social environment. Testosterone is produced in the testes and is related to sexual behavior and sperm competition. Corticosterone is known to be a main regulator for energy metabolism and social stress responses. In our experiment, we investigated how male birds adjust their behavior and hormone levels to experimental variation in sperm competition risk. We use zebra finches as our study species because they are a key model organism in research on sexual selection and sperm competition. At first, we assigned males to a partner and let them form a pair bond. After 2 months, we randomly allocated these pairs to one of the two social treatment groups. In the double pair group, our focal pair was kept with another breeding pair, which resulted in elevated sperm competition risk. In the single pair group, the pairs stayed as they were without any sperm competition risk. We provided next boxes to induce breeding of a first clutch. We replaced the eggs by dummy eggs and removed it after 2 weeks, which stimulated the birds to produce a replacement clutch. Our data collection was synchronized with regard to the laying of the replacement clutch. We observed male behavior before and during egg laying of the replacement clutch and performed a standardized intruder test. To measure hormone levels, we took 2 blood samples, one before and one after the start of the social treatment. Here you get an impression of our setup. Cameras were attached to the cages, so we could film several replicates in parallel. Now I will show you some of the results, and I will start with the behavior observations. We quantified male song durations as a measure of courtship before and during egg laying of the replacement clutch. First of all, we see an increase in song duration in the progress of the breeding cycle. Surprisingly, males sang more after the female started egg laying. In contrast to our prediction, we found that males from the single pair group sang more than males from the double pair group. During the 10 minutes intruder test, we recorded the total time the focal males spent in aggressive interactions with an unfamiliar male. Also surprising for us, we found that males in the single pair groups spent more time in aggression than males from the double pair group. For the hormone measurements, we took 2 samples per individual, one before they entered the social treatment and one during the social treatment. Before the social treatment has started, there was no difference in testosterone levels, which makes sense because all of the males were kept only with their partners in the same social condition. Testosterone levels dropped significantly during social treatment. When comparing the 2 groups, we found that double pair males have higher testosterone levels than single pair males, which was also in line with our main prediction. We found no difference in corticosterone, neither between treatment groups nor between measurements. To conclude, we found that in contrast to our predictions, males showed reduced courtship when encountering sperm competition risk. This could be explained by a larger number of birds and thus more interaction partners in their close environment. Then males invested more time in behaviors for example, like nest defense, which was also observed in the video recordings. Additionally, also in contrast to our prediction, males from the double pair group spent less time in aggressive interactions with intruder males. This could be explained by the fact that males from the double pair treatment have experience with other males in their close environment after breeding together with another pair for several weeks. And thus, they may reduce the cost of escalated aggression. Regarding the hormone levels, we found that testosterone levels in the double pair males were higher than in the single pair males. This was in line with our prediction. Furthermore, testosterone levels decreased during treatment, which could be explained by the fact that male zebra finches participate in incubation and downregulate their testosterone levels by that time. Regarding the corticosterone levels, we found no sign of physiological adjustment to the social treatment. Overall, we found that males adjusted their behavior and hormonal phenotype in response to the social environment, but not always as we originally thought. But whether the risk of sperm competition is the main driver that explains our findings, or if other factors influence the male phenotype, is not completely clear. With this, I would like to thank my team and all the helpers. And this is a pre-registered study. And if you're interested in more information about it, just follow the OSF link or contact me for details. Thank you for your attention.